such analysis to understand and characterize these variants can be performed at many different levels to investigate the functions of variety of different genes in multiple different research areas. Right? These research areas can include drug metabolism by studying the enzyme function that uh, mm, that is key in the catalysis and the key in the functionality of specific metabolite. And this can also end up trying to characterize, we can also end up trying to characterize mutations in specific genes that can cause tumor. In that case, genes that are linked with tumor progression and tumorigenesis. And we already spoke about sickle cell anemia. This in case, this in this example is caused by the effects of germline mutations that are inherited from our parents. So furthermore, one can also use genomics to explore pathogens and to understand important properties of their pathogens as a result of their, or as a function of their genomic variants, to study virulence, to study disease transmission, and to study drug resistance, etc., etc. Many of these examples will become clear as we review several project and exemplar projects as we go along in the sessions, as we go along in the program. And some of this we can also discuss in the upcoming slides in today's session. So if you have any questions, please let us know in the chat box. We will discuss that before going to specific examples that will help us understanding the genomic variance analysis. Okay, if there are no other questions, let's proceed. So until now, we have tried to understand the concepts and the types of variants and mutations and how to, for example, characterize them. So let's understand why and how these mutations, yes, Akshita, this will be available. So let's understand how these mutations and variants play very important role in the progression or development of cancer. Or better, can we say, how can the mutations will actually help in tumor formation? So we already know that there are various important genes that play key roles in cell cycle and regulation of cell growth and etc. So any disturbances in these genes will actually result in dysregulation of cell growth and eventually enhancing the development of tumor. So if we focus on those genes and focus on variations on those genes which could result in tumor development, we can actually discuss a couple of examples. For example, these genes are actually generally termed as tumor suppressor genes because usually these genes regulate the cell growth, and if they for example, if they encounter any defects in the cell growth, they end up inducing apoptosis and hence saving the eventual progression or eventual progression of the tumor and development of tumor and progression of the tumor in these samples. But if these tumor suppressor genes get mutated and, for example, get inactivated, it will or the mutated version may end up altering the normal function of these genes, then the regulation will be affected and eventually that could result in uncontrolled growth of these cells. So some of the examples of tumor suppressor genes that you can easily relate and you can actually find everywhere in the biomedical research area is the TP53 that we all know about, and P10 or some of them. So these genes that are responsible for cell growth and potential mutation that is caused in these genes may result in promoting, for example, promoting tumor are termed as oncogenes. And there are also some of the genes that are responsible for cell growth, and if any mutation in these genes that are responsible for cell growth can be related to promoting or accelerating, or actually amplifying their normal function. But that was the word that I was looking for, amplifying the normal function. And these genes at their normal level that promote cell growth, but at their amplified level can promote abnormal growth, are termed as oncogenes. And any mutation that results in uncontrolled cell growth will again result in tumorigenesis. And we can actually find some of the examples of these oncogenes that are HER2, which is epidermal growth factor receptor, human epidermal growth factor receptor, HER2. That is one of the oncogene. And some mutations in these genes will actually result in these receptor binding to a lot of growth factors and promoting abnormal growth of the cell. And there is also another oncogene, which is epidermal growth factor themselves, right? EGFR, epidermal growth factor receptor themselves. And this again will bind to many growth factor molecules and promote tumor progression and tumorigenesis in general. And there are also other forms of genes and other genes which are involved in DNA repair mechanisms, for example, and any mutations in these DNA repair mechanism involving genes may also end up changing the characteristics of the cells to a malignant level. So some of these mutations can be acquired, which is the most common cause of the cancer. They can be acquired due to stress. They can be acquired due to many times copying the DNA code to several generations, and they can also be acquired due to random chances, right? And some of these mutations can also be a result of hereditary cause, or these are all termed as germline mutations, which are less common. And the genes that are famous for germline mutations are the BRCA genes. So if specific mutations in the BRCA1 and BRCA2 genes get propagated from the parents to daughters, or from the parents to cells, from the parents to progeny, then that will result in most likely, or result in a higher risk of developing breast cancer and ovarian cancers, etc. So some of the mutations that are acquired, for example, may be concentrated in several different portions of the gene, and these are all somatic mutations or acquired mutations. And those mutations which are known to cause tumorigenesis or tumor progression or cause cancer are termed as driver mutations. And those mutations which are known to be associated with the presence of cancer, but their clear relationship with the cancer development, if that is not known, then those mutations are called passenger mutations. And when one discusses, or when one focus on one specific gene, for example, TP53 gene, which is very well known for the protein that is tumor protein TP53. And this protein, as we already discussed, acts as a tumor suppressor and regulates the cell division by keeping the growth and cell division under control. So the mutations that are present in TP53 are of numerous type and also numerous variety. And you can actually check, right? If I did not butcher your name, you can go to repositories like Cosmic and mainly Cosmic to study potentially pathogenic somatic mutations related to one specific gene. So if you type TP53 gene in Cosmic, then it will end up listing all the known variants and mutation that is known to cause that is known to be associated with the tumor development themselves. So there are also other forms of annotation in the Cosmic database, which will annotate the mutations to be potentially a driver or potentially passenger or unknown causes, etc. So this TP53 gene themselves, it is implicated in many cancers, not only in specific type of cancer, many cancers, and it helps, as we already discussed, it induces apoptosis, and when the protein actually detects some abnormality in the genes, and several of the mutations that is present in the specific regions that bind and recognize DNA code can lead to be promoting tumorigenesis by not controlling the replication of the cells with damaged DNA. So this protein will bind to DNA and will identify incorrectly translated replicated bases, and when you observe several mutations in the binding site of these genes, that will actually that are actually usually associated with tumor promoting driver genes. And there are also other variations or other variants and other mutations that are generally observed in the structure of the TP53 genes protein, which may not be directly related to their functionality, but one has to say, or one has to keep an open mind that we have not completely understood the effect of those mutations until now, even though we have understood many different mutations and their involvement in tumor progression and tumorigenesis for TP53 gene in specific. So the relationship between disease-causing mutation is not limited to cancer. In fact, this relationship and prevention has developed a lot, has been, for example, worked upon in a lot of other diseases compared to cancer. Like one of the very well-known story is about cystic fibrosis, right? Cystic fibrosis is as we know, can be understood as it causes chronic lung infections by the changes in the mucus secretion. So it is usually or generally caused by mutations in one single gene, that is CFTR, cystic fibrosis transmembrane conductance regulator gene. So this mutation can actually result in either no CFTR protein being produced, or some of the proteins that are produced may be ill-formed or malformed, and they cannot perform their key functionality in the cell. And there are, depending upon what happens, or depending upon the variety of different variants, there are 5 classes of these CFTR mutations, and they can occur in different stages, from the protein synthesis to producing insufficient protein. The most common disease-causing mutation in all these 5 different classes of CFTR is this protein processing or maturation processing protein, where this one specific amino acid, phenylalanine, is actually deleted. And this actually occurs in more than 80% of the patients, and that causes incorrect regulation of the mucus formation, and hence it results in chronic lung disease. There are also other mutations like the one that is in the gating, that involves the gating mechanism, like glycine to aspartate, and which causes complete loss of the protein in the plasma membrane by resulting in the gate being too narrow. So generally, the life expectancy of these cystic fibrosis patients can be improved or can be lengthened when we diagnose these mutations early, and when some of the symptomatic treatment therapeutic regimens are produced. But these are actually results of hereditary mutations, right? Or they are germline mutations, and one can observe the presence of these mutations when we look for these specific mutations from the development stage of the progeny themselves, right? So depending upon where these mutations occur, as we see that there are several advancements in in understanding these CFTR mutations has been made, and specific gene modulators for these specific mutations are also developed, and these modulated drugs have the ability to, for example, restore partially or fully the functional expression of these specific CF causing mutations, and they also have been classified into addressing these specific mutation separately or accordingly. So this highlights the importance of studying genomic variants. No, I mean, yeah, that's a great question, Komal. Why only 39 mutations of CFTR is considered for modulated treatment? Why not the other 1,700 mutations? So we can only act upon those mutations for which we can successfully generate or develop modulation. There may be several other mutations, and many of these mutations may remain uncharacterized or may not be reachable themselves. The modulators are also therapeutic drugs that has limited reachability or that has limited functionality. One has to identify, or one has to use only certain targets of these certain target mutations of these CFTR. No? So that is why people are still working on these modulator productions, and CFTR is very actively developing research area. Understanding CFTR mutations is very actively developing research area. And one has to keep saying that these modulators will only partially, may only partially restore the functionality of the CFTR, and several more research has to be successfully developed and successfully performed to generally increase the quality of life of these patients. Until now, we have only succeeded in extending the life expectancy of these patients, and eventually when we end up in improving the quality of the life, then people can actually consider this as a success story. But nonetheless, understanding the mutation at several different molecular level will actually help us generate specific intervention for those specific variants or mutation, and can actually help the help treat patients suffering from these deadly diseases in this condition, right? So mutations classified at, for example, categorized as at single nucleotide positions are not the only cause of disease, not the only disease causing mutation. For example, in the case of, I'm not a professor yet, Komal, thank you. So in the case of Huntington's disease, which is a rare inherited disease, we already know they cause for example, progressive breakdown of the nerve cells in the brain. And this has a wide impact in person's functional abilities, and it usually results in movement disorders, usually result in cognitive disorders, and may end up resulting in psychiatric disorders in the end. For example, we know that Huntington's disease is an inherited condition where instead of point mutations that we have been discussing so far, a specific gene on the chromosome 4 has an abnormal number of CAG codons. This is an example of duplication mutation that we discussed, or that we just touched upon earlier. So where the CAG codon is actually multiple times reproduced or multiple times duplicated in the HTT or Huntington gene. So this gene encodes a very large Huntington protein, and these additional repeats actually change the structure of the protein completely, and this is the result of progressive movement disorder or dementia, behavior abnormalities, etc., etc. For example, the Huntington diseases typically emerge in around midlife, 45, 55, or even later at that point, but there are certain cases, and there are specific cases where juvenile onset has been observed, and when we can, if we can diagnose the presence of these characteristics motor signs, based on the presence of these characteristic motor signs, then we can look for these mutations in those juvenile onset cases. That is what is being done by the Huntington Consortium in 2015, which published a study of over 1,000 individuals with early and late onset of this disease, and by by understanding the whole genome sequencing and understanding the targeted sequencing of specific chromosome 4 sequencing, researchers discovered very important regulatory mechanisms that are differentiated between the groups for which we observe early disease or early onset, and for another which we observe late onset. And that actually is associated with the chromosome 15, where we have abnormal copy number variations of a specific gene, and that results in the slower development of the of this specific disease progression and etc. of different mechanism of disease progression. And this chromosome 15, for example, this specific gene in the chromosome 15 that have abnormal number of copy number variation can actually help us identify mechanisms of this disease progression and help us develop therapeutic therapeutic intervention. So now what I was actually trying to say is the chromosome 4 is what is associated with the development of the Huntington disease. But when they perform this targeted sequencing effort in these 1,000 individuals with early onset and late onset, their differential variant analysis resulted in identifying specific regions of chromosome 15 that had higher number of copy number variation or higher number of copy number aberration. And for them, right, those for those who contain this higher number of copy number aberrated chromosome 15, they are associated with the slower development of this Huntington disease. And by understanding this variant in particular, one can identify mechanisms of this disease progression and relate that to the therapeutic intervention by naturally understanding the disease regulation and etc. etc. That is what this study actually helped us understand in specific. So where without any intervention and natural variation that occurred for a specific group of people acted as a potential regulatory mechanism for a deadly disease, we can exploit to treat the specific early onset of Huntington's disease. So proceeding further, we can actually turn our focus into pathogen genomics data analysis. In this case, we already know tuberculosis is so difficult to treat because they usually attack the lungs and they can spread to other parts of the body like brain, spine, etc. And this is caused by Mycobacterium tuberculosis. Why is this so important? In the 20th century alone, tuberculosis was the leading cause of death in the United States, and today most cases are usually cured with very difficult to follow and very complicated antibiotics regimen, right? But in the last 3 years or 4 years, researchers have actually discovered that this bacteria has become more and more resistant to not one, but several different types of antibiotics. So it turns out that this resistance can be associated with a specific set of mutations in specific genes or their promoters. So understanding which gene and which antibiotic that gene mutation, resistant to which antibiotic that gene is associated to are very important in understanding the development of resistance in general. So there are also large, for example, part of developing new drugs to address this surge of multi-drug resistant tuberculosis infections themselves. And in addition to this multi-drug resistant tuberculosis, there is an existence of extensively drug resistant tuberculosis, which is resistant to all different types of antibiotics or most of the different types of antibiotics that it has been tested with. So there are multiple studies that focuses on understanding the population level or population percentage distribution of these multiple drug resistant and extensively drug resistant tuberculosis from single infected isolated area. And there are also studies, for example, in as you will know, in some of the example projects that we have, study 22 years old study performed in single patient to understand the development of drug resistance to variety of different drugs that are used to treat that specific patient. So we have more examples, like examples in Ebola virus, where we know that the viral protein VP24 is a nucleocapsid protein that is associated with inhibiting interferon gene expression and interferon mediated antiviral response. So this actually prevents the nuclear import of the signal transducer and the activator of the STAT1, and hence the interferon mediated antiviral response is heavily depleted in the Ebola virus affected infected patients. But several studies on specific strain of Ebola virus that exist with reduced transmissibility and reduced disease pathogenicity or virulence actually identifies specific mutations at residues 142, 146 that are able to, for example, reduce the activity of the VP24 by 90%, right? So these mutations that are naturally occurring were the result, or were the were one of the starting point that ended up helping us develop vaccines for this Ebola virus in this specific epidemic at 2014, 2016. So there are other examples as well. For example, this actually discusses the use of understanding the rhizosphere, which is defined as the soil zone around the plant roots, right? And this soil, as we know, is rich in nutrients, and in this region there is an intense interaction that takes place between the plant root tips and plant roots and the microbes that are present in the soil, microbes that are present in the plant roots, right? These microbes inhabiting area are generally termed as rhizobacteria, and they can actually regulate all the known biogeochemical cycles of this specific space, and they can regulate degradation of organic chemicals, and they can actually preserve the components of the soil chemistry. And understanding the mutability or mutation of the, for example, mutability in all the positions of this rhizobacteria genome can actually help us understand the variety and variation that is present in several different rhizobacterial genome in specific. There are a couple of publications that are involved in isolating specific rhizobacteria genome and in a genome-wide association studies, GWAS-like studies in understanding the heterozygosity of this specific rhizobacteria, which we know are very beneficial to maintaining the healthy ingredients of the soil biosphere. People actually characterized mutations that are beneficial to maintaining these interaction at a healthy rate, and some of the mutations that are known to cause depletion in the functionality of the rhizobacterial genome in general. So that is also possible. So this is not only limited to, for example, understanding the bacterial genome that inhibits or that are inhibited of the inhibited of the soil rhizosphere. One can also use general genomics data analysis in promoting crop yield and in promoting understanding the genetics of agriculture, which precedes the use of genomics in agriculture by several decades. So the efforts in those areas are also heavily advancing, because generally when you see the amount of data that is available for us to compare our samples with are focused more on human genome and disease causing human genes, and less on general crop yields and crop production and agriculture in general. So in those aspects, people focus on not comparing the genome with the reference genome. If the reference genome is not present, then they work out, or then they perform GWAS-like studies in trying to sample variety of different genomes and understand the variation that is present at different levels inside the plant sequences, etc. So this is not limited to only plants. This can exist at different levels. Also in metagenomics levels, also you will get deep and detailed understanding of these applications when you go through some of the associated resources that is listed in Introduction to Bioinformatics, and sometimes bytes and molecules in the resources. So with that, I will conclude today's lecture, and in the next following sessions, we will focus more about the important part of these studies that are related to our ability to compare the DNA code from given individual to a reference. Right? This comparison is actually made possible through the computational advances that will take millions of these short reads and align themselves to the reference to extract meaningful insights, as we have been discussing so far. So the next session is to understand the basic concepts of sequence analysis through performing the sequence analysis in an environment like R. So you will start with understanding what is a DNA sequence string and why we need to compare it with the reference sequence, and what do we gain by comparing it with the reference sequence, and what are the variants that we can actually characterize. You can perform this alignment yourself, and what are the different types of alignment that one can focus on, and how to do a test alignment in R's case, and how to do similar analysis by uploading files in our Taubot Bioinfo platform and visualize the output in JBrowser or IGV, etc., etc. That will be our discussion for the next session.